So it dawned on me, like right after I finished saying what I was saying, that we hadn't discussed how it is we actually protect people from viral infections. And the most promising, at least in the course of your lifetime, has always been the use of vaccines, where you present live attenuated virus or killed virus or molecular subunits of viruses into the host organism, meaning you, and you let your specific immune system identify it, meaning your T cells and your B cells figure out how or what piece of the virus it's going to attack, and then it will attack it. That's all it does. It usually uses what's called matrix compatibility 1 complex mechanisms to actually do this. There was a discredited discredited paper that came out that said that the materials that were used to create viruses, or I should say vaccines against viruses, created different disease states. And it turns out, of course, all of that is false, meaning the materials that are used to stabilize the attenuated live virus or the killed viruses or the molecular subunits, all of those experiments were fabricated. And unfortunately, it's created a subcontext in our fight against COVID-19 at this point in time, where people believe that because they don't feel like they're sick, they don't get infected, or they're just simply not smart enough to understand how it all works. That's not to say that they're not smart people. It's to say that somehow, because they haven't critically analyzed all of the information that says, hey, bad things will happen if you get virally infected, they decide they'll take the chance, and that's their right. There's a reason why you're being asked to wear masks. It's not just to protect you, it's to protect the other people that are around you. Vaccines are designed to protect you from becoming a meal of that viral infection. When we're thinking about the last idea that I wanted you to sort of contemplate here, and the reason I almost forgot about these things, not because I didn't want to talk about them, but because I'm like, okay, how do they actually fit all of this information in? Because before we were talking about DNA and RNA, well, what happens if there's proteinaceous infectious particles? Very small structures with no nucleic acids. They just have proteins, and they cause fatal neurodegenerative diseases. You may have heard of mad cow disease, Jakob-Creutzfeldt or Creutzfeldt-Jakob disease in humans, Kuru, which was my favorite when I was growing up as a kid, where specific tribes in the Polynesian islands would spread the disease state by eating the brains of their victims. So you would actually eat, not necessarily the brains of their victims, it could also be, I remember at one point in time reading about ceremony at the end of life. So it could be that they were just eating tissues from other humans, and they were capturing Kuru, scraping inside sheep. And around here, what we actually see inside a lot of our deer is what's called chronic wasting disease. So somehow, some small proteinaceous particle is being passed from cell to cell, creating different examples of each of these. Bovine spongiform encephalopathy wiped out entire herds of cattle in Europe. It's wiped out entire herds of cattle inside the United States. We don't tend to hear about it as much here because to take care of it, you literally had to kill everything. You're going to see the same thing probably happening in the Northeast with deer. Not so much the moose and the elk inside the Northwest are where those are happening. But what I want you to leave this class with is this. Normally we get rid of things like viruses by heating them and injecting them into humans and going, hey, let's let your immune system take care of that. Notice prions destroy nervous system tissue, and they're not destroyed by heating. So when we used to teach you, or when we do teach you, that the primary, secondary, and tertiary structure of proteins are what allow it to become effective. And when you superheated them, or when you heat them and you cook them or something like that, you change the three-dimensional structure. The three-dimensional structure does not change the prion's effectiveness at becoming a transmissible, fatal, neurodegenerative disease material. One is just fascinating if you think about that. So we'll finish off the semester with that. I think if I do have a couple more ideas, I'm going to turn them into small things I'll put up on Blackboard for you. I'm going to put these up on YouTube right now, and I'll hopefully speak with some of you tomorrow. Hopefully some of you have your power back. Be safe. Bye. Oh, here they are right here. That's kind of cool. Endogenous PRPs, converted accumulation. These are the accumulated materials that are actually causing the disease states inside of the brain. Okay, cool. Bye.